Thank you for introducing me. I will present the work I performed as a postdoc at the Vrije Universiteit Brussels under the supervision of Professor Dominique Maas with the collaboration of Microflow Group, which is run by Professor Wim de Molsch. Currently, I'm in Warsaw. I'm working at the Biosense Lab. It's a startup who supports my presentation today and is also giving me affiliation for the talk. So first, I will start with a brief introduction about protein self-assembly. As you can see, looking at the endpoint of the protein self-assembly, we can distinguish different characteristics, different morphologies, like amorphous aggregates, crystals, oligomers, or fibrils. And in my talk, I will focus on ordered structures, and the ordered structures are crystals, and there you can find a pattern in how the single molecules are ordered within the 3D space. And fibrils are another example of ordered structures, because there you can find the pattern, how they are ordered and aligned within one axis. So sometimes we can say that the crystals are ordered in 3D and fibrils are ordered in 1D, in one dimension. For the crystals, the proteins and the homogeneous solutions, they self-assemble in a manner that one single molecule does not overcome any structural transitions. So when we have a crystal structure of a protein of our interest, we can, based on the crystal and investigation with an X-ray, we can solve the structure and we can see how much of beta sheets are there, how much of alpha helix, and with a very good resolution.
I'm so sorry because I can see on the chat that people can see your slides, and now I realize that we see only your conclusions. I don't know what you can see, Nicola.
I see the presentation. You don't?
No, we see only conclusions.
Okay, that's strange. I see.
I realized that probably that maybe you see all of the slides. So this is why. Okay.
Well, everyone can see only conclusions.
Now we can see protein self-assembly. Are you sure that have you shared your entire screen or only the window? Because if you shared only one window, it can be shared only for PowerPoint, not for the presentation.
Yeah, I did share and only no, I did share the PowerPoint.
Okay, so this is right. Because if you share whole screen, then we can see the presentation. Because now we see only PowerPoint.
Entire screen.
Yeah.
Okay, and select and share. And voila.
Oh yeah.
Can you see it now?
Chat, can you? Yeah, Aaron said that.
Okay.
Chat is missing. Sorry for interrupting.
Okay, thank you, Mikołaj.
Oh, thank you for the remark. So if you didn't see the slides, now I can make it up for you, and you can see all the structures presented in a scheme. And so I was talking about crystals and fibrils as ordered examples of proteins aggregation, and which I will be talking about today, and how they transit from the single molecules towards the organized structures, how they do self-assemble. I think it's a very nicely put on a scheme analogy for the energy landscape of these transitions. When we look at the single protein folding process, I think most of you have seen this scheme, this energy landscape. Then this folding protein overcomes some structural changes that will locate it in some energy minima that are very comfortable for the protein to stay at, because they are minimum of an energies. And basically the same happens when more than one molecule comes together with another one of protein, and they form structures. So the structures that has been defined in the former slide are at the energy minima. So we define them because they are stable and we can observe them with methodology we can have in our laboratories, for example, with the AFM, like oligomers or amyloid fibrils. And today I will focus mostly on fibrils. And for the self-assembly pathways of fibrils, we can distinguish mainly the two pathways, where the first pathway endpoint are braided fibrils, and their characteristic is that they are flexible and they can intertwine with each other, and to form they usually have a transition forms like a large oligomers or small oligomers. They go through this pathway, and the beta sheet enrichment here is rather small, while for the other pathway, which results in formation of rigid fibrils, the enrichment in beta sheet is typically higher compared to the other pathway, and the transition points for this pathway are smaller prefibrillar nuclei and rod-like, very straight and rigid protofibrils. But in real life, typically, when you will have a random system, so I mean like a random solution of your protein, and it will start fibrilizing because of oversaturation, you will have a mixture of everything. So what you see here is a very idealistic distinguishment between all the processes that are happening in one typical solution. And so looking at that distinguishment, we can understand that formed fibrils can have different morphologies. Like here, the braided fibrils or rigid fibrils, they have different morphology, and it's like with crystals. Everybody knows that crystals are polymorphic structures, so they can have they can be composed of exactly the same monomers, but organized in a different way. And what's the consequence, and why it's important to understand and to investigate the endpoint results of the morphology of the fibrils. So fibrils are hallmark for neurodegenerative diseases, which example is Alzheimer disease or Parkinson disease. And it has been shown that patients who did have very severe symptoms for Alzheimer disease, they did have fibrils in their brain. And at the same time, they did find people who were healthy and they did not have fibrils in their brain. What the reason could be? So it's been still under the investigation, but there are arising proofs that how these fibrils are structured influence their cytotoxicity because of their spatial organization, how hydrophobic parts are exposed to the surface or hidden inside will determine whether they have higher or lower affinity towards the cell membranes. And important information here, fibrils in both cases, in Parkinson and Alzheimer disease, they are located outside the cells in the extracellular matrix. And what I will talk about today is how gravity levels can influence the selection pathway for the formation of a different morphologies. So this is a little bit of a theory still. I took a very scheme of a single crystal, and let's assume that the single crystal is being built. Upon growing, this crystal recruits in a very close proximity small native proteins, making a depletion zones. So the liquid in the close proximity around this crystal is depleted from the single molecules, while the liquid further away has a higher concentration of these molecules. As a consequence, the density of the liquid close to the crystal is different than the density of a liquid further away. And once we have a density differences in one system, the convection phenomenon will occur and it will mix it all together. The density differences convection process is strongly related with the gravitational force. The higher gravitational force, the mixing is more efficient. So the actual concentration in a close proximity of a growing self-assembly structure will be experienced as less dense in smaller gravity or high dense in a higher gravity. And another effect that gravity will have in the self-assembling system is when the oligomers will nucleate and grow. At some point, the oligomers, as I've shown, they can be a transition state towards formation of braided fibrils. At some point, they will be heavy enough to sediment, and they will sediment across this depletion zone, and they will find on their way more and more single molecules to recruit and grow with them further. So the higher gravity, the smaller oligomer to already sediment and grow. And now I will present the results I obtained during my research. I employed a lysozyme solution at 3 different concentrations. They were all supersaturated concentrations, 12, 20, and 40 milligrams per milliliter. And I divided it into 4 groups. One group was left without any agitation, so that was my quiescent control. Another group was exposed to 20G hypergravitation. Another group was exposed to 200G hypergravitation, and the last group was exposed to shaking. And the shaking is a very classical approach to form fibrils. And typically when you want to produce some fibrils in the lab, you will use orbital shaking, as I did in my experiment. After 2 weeks of such an exposition, every sample has been investigated with the AFM. And here you can see the comparison of all the sample class. And first, we can immediately say that in the quiescent conditions, no fibrilization occurred, which is very apparent. Here we can see just a small oligomers. At 20G, we can see that fibrils were formed as well as 200G. And in shaking, also the fibrilization happened. But there are very huge and aberrant differences in the morphology between the fibrils formed in the hypergravity and between the fibrils formed in shaking. In shaking, the fibrils...
Marzena, sorry for interrupting, but you have 1 minute left, just to let you know.
Okay, I will be quick. And in hypergravity, the fibrils were flexible and very long, which I could do some statistics based on the AFM images. And the results were valid, statistically valid. The differences could be statistically valid. And also I performed on the endpoint solutions infrared studies. And these studies did show that samples from shaking, they have a huge contribution of a beta sheet, both for like an entire sample, also for the sample split it into two. So when I filtered the sample with 0.1 micrometer filter, I had a solid part and a liquid part. And then both solid and the liquid part for samples from the shaking had a largest contribution of a beta sheet, which was not the case for the remaining samples, both for hypergravity and samples from the quiescent conditions. The samples from hypergravity and from quiescent conditions, they preserve, like generally speaking, the native conformation of a protein. And this can indicate a very different self-assembly pathway between the two groups of samples. And it is also the very first proof that this studies has been published that just by applying a hypergravity, we can differentiate the fibrilization pathway for protein solutions, and that the hypergravity can be a fibrilization trigger. And thank you very much for listening.